next speaker is Magda Markowska from the University of Warsaw, and she's talking about CONET, copy number inventory model of evolutionary tumor history for single-cell data.
Yes, hello everyone. My name is Magda Markowska. I'm a PhD student in Ewa Szczurek's lab at the University of Warsaw, and as you can see, today we are coming back to deconvoluting evolutionary tumor history. And this time we will deal with the copy number aberrations, not with SNVs like it was yesterday. I have to go. So copy number alterations are ubiquitous across all cancer types. They are also the most common alteration type that is associated with tumor hypermutability, and they can drive tumor evolution by deleting tumor suppressors or amplifying oncogenes. Recently, new technologies have enabled us to obtain a relatively even coverage across the whole genome. So we can call single CNV profiles for single cells. But so far, no methods have been published for inferring copy number evolution for this type of data. Yeah. OK, so maybe a little bit more about the data. On the left, you can see the histogram of corrected counts for a breast cancer sample of 260 cells, single cells. This data we call the per-bean data because they are binned into quite wide genomic bins. In this case, they were 150 kilobases wide. And you can see that this histogram looks more or less like a mixed normal distribution, which is plotted in the green. And we can transfer this per-bean data into the per-breakpoint data. And in our case, these are the corrected count absolute differences in adjacent bins. They were here divided into two subgroups. So the red subgroup are the corrected count absolute differences for loci where we do not expect a breakpoint. So we don't think there's a copy number change there. And obviously, the expected value for this truncated normal distribution is 0. And then the blue part are the differences for the loci where we expect breakpoints. And this is also a mixed normal distribution with the most prominent component being equal to 1. And so this is quite obvious from the histogram of the per-bean data. Probably we had deletions from copy number 2 to copy number 1. OK. Here you can see the overview of our model. In the panel A, we have the data preprocessing steps, which are, of course, quite important. The color dots signify single cells. And the shapes, the gray shapes, they signify genomic loci. And first, we need to correct the data for GC content and mappability and normalize it. In this way, we obtain the per-bean data, so the corrected counts in genomic bins. The cells are in rows, and the genomic bins are in columns. Again, we further transfer this data into the per-breakpoint data. So this time, we have in columns genomic loci, not genomic bins. And the cells are still in the rows. The last step of the preprocessing of the data is we establishing the set of potential breakpoints. So we just need to say which breakpoints we consider to be part of copy number events. And this is more or less the user's decision. The more breakpoints we include here, the longer the modeling step can be. So in the panel B, we have the most important modeling step, the heart of our CONET model. First, we can see an exemplary CONET structure. We have a healthy cell as a root of the tree. And then the tree is not a non-binary tree, so it's branching, and each node signifies one copy number event, which is denoted as a start breakpoint and an end breakpoint. This is not a phylogenetic tree. And we can see that the copy number events can overlap. As for example, maybe I can point here, we have a short event from a diamond to a circle. This is, this doesn't work. Oh, OK, I get it. here, and then the longer event that is from a star to a triangle. And also the breakpoint loci can repeat in the tree, both as a start and end breakpoints. The second part of the model are the model parameters. So these are those two distributions that I have described before. The corrected count absolute differences distribution, one for when our model is telling us that there is no breakpoint. This is the black distribution, and the red one when we expect a breakpoint. And we use those distributions to calculate the likelihood of our CONET. And at the bottom, you can see the summary of our probabilistic Bayesian model, where we use the differences to calculate the likelihood, but we also use the per bean data in the form of corrected counts for regularization.
So just to understand, so the tree, does it have breakpoints in it, or is it actually the copy number sequence itself?
No, these are only breakpoints. We only say that from this point to this point, there was a copy number event. That's it, nothing more. We don't say by how many, because that vastly increases the space of possible trees, which is already huge.
And they could be overlapping, so it could have...
Yes, they can be overlapping. Yeah. Yes, exactly. Yeah. So we have designed a very efficient MCMC sampling procedure, which returns the best tree structure. And afterwards, we can attach the cells using maximum likelihood attachment. And the cells can be also attached to internal nodes. With that, we can call final breakpoints in each cell. And also, we can perform final copy number calling when we cluster the beans that have underwent exactly the same copy number history. And then we just calculate the average corrected count and then round it to the closest integer. And all the beans that were not concerned, like they were not inside any of the copy number events, according to our tree, we say they have a copy number 2. Without checking.
What's EVO here?
Sorry?
How do you distinguish EVO from a deletion? EVO, allele dropout.
We don't consider allele dropout at all.
Single cell. Most methods, you need to amplify the DNA. Not in all methods, but with the methods that people use, you amplify the DNA and you have a dropout, which will look like a deletion, right? You won't have reads in this region.
But these are only corrected counts. We do not consider the... also, the data we use has relatively even coverage, and they don't use the pre-amplification step in their methods.
We don't look at SNVs at all.
And we don't care about SNVs. This is
on the size of, yeah, but because of the size of the beans, you think are larger than the...
So is it DLP plus data?
Yeah. Yeah.
Okay.
Yeah.
So you're assuming that there's no amplification that disturbs the...
Yes.
Is that true, or...
What
Is the
about the bins? Are the bins in the
how big are these bins?
The copy number changes over the tree?
We do all the likelihood over the breakpoint.
So you might miss a break
We-
because of an ADO, right?
Because of what?
I don't see the point of the ADO in this data.
I'm trying to ask things too.
Okay, sorry, yeah.
Yeah, I know, but I just didn't hear. What? Yes.
What about breakpoints? Are they matched together, and what independence between them? Because
Yes. So we basically...
it has 2 breakpoints and they go together.
We basically consider each breakpoint separately, and we say that they are independent. And we check the probability of each breakpoint using those two distributions. So if our model says that in this cell it sees a breakpoint, then we use the red distribution and check the PDF function for a corrected count absolute difference in this specific locus.
So at the breakpoints, the flanking regions can change the copy.
Yes, exactly. Yeah.
Essentially, the model tells you what
That's
is
the-
the set of breakpoints that your cell should acquire, depending where it is attached side.
Yeah, that's basically what we do. Yeah, we just check the changes and see whether they indicate a breakpoint or not. And then we, because the tree is very complicated, we have many breakpoints, and we also marginalize the cells attachment and just look for the tree that returns the highest likelihood.
So maybe one last question. When you have the red distribution,
Yes?
said you model this as a mixture of Gaussians, or is it just a single Gaussian?
No, it's a mixture. This one's
Do you have a method to determine the number of components? How do you
We
do
check the number of, we sample the number of components during the MCMC sampling
So it's
procedure.
like, is it a Dirichlet process with because how do you know how many you need? You could have like lower breakpoints on quite a lot.
Yeah, we say that I think we start from 5 and we either decrease or not. We don't want to, because basically they should be either 0, 1, 2, 3, and then we don't we see that the higher ones are not that important, so we don't model that. Okay, so maybe let's go to the results. First, for the simulated data, here you can see the results for breakpoint detection. So here we could have compared also to a method that only perform copy number calling. The pink one is CBS plus merge levels, and the green one is HMM copy. And then we have CONET in 3 different versions. They differ by the method that we have used to obtain the potential breakpoint set. So it's either HMM, CBS plus merge level, or we give our model the true potential breakpoint set without saying which breakpoint is in which cell, but just a set. And for the SciCone, this is the other model that is also a preprint, and it also has copy number events that give the beginning of a copy number event and an end of copy number event. And it also has 2 variants. First, the one when they use their own method for detecting breakpoints, and the other one, similarly to CONET, where we input the known potential breakpoint set. And you can see here that both for a false positive rate and false negative rate, in terms of breakpoint detection for different simulation scenarios, so with lower noise and higher noise and different number of cells, which is indicated at the bottom. Basically, CONET performs best, and the performance of CONET here depends less on the method of detecting breakpoints at the very beginning. And
when you say known, this is actually you put the real breakpoints there.
Only the set. So we only say, please only take this loci, this, and let's say 3 loci, but we don't say in which cell. Only the ones that we are.
But it's the ones. It's basically sampled from the true set of loci.
Yes.
It's not like. Okay.
Yeah.
So you assume you know that somehow. I don't know.
Just to.
We have a method which well recognizes the candidates.
And also then we can fairly compare to SciCone, right? Because we start with the same set of potential loci. Okay, and here's the results for the tree inference. This task is much more difficult, and here we could have only compared to SciCone. The setups are basically the same. And in terms of edge precision, CONET varies from, let's say, 50 to almost 100 in the lower noise. And with, with edge precision is up to 75% for the lower noise, and up to also 75% for the higher noise. A little worse when we use HMM copy to infer potential breakpoints, and the differences here are bigger when we use different methods for detecting the breakpoints. And for edge sensitivity, the results are similar, and always CONET performs better than the other method. And last, I would like to present to you the CONET inferred for this DLP data set of 260 xenograft breast cancer cells. Basically, this is much more complicated than the example. We have inferred around 200 different copy number events, and they were collapsed whenever it was possible, so when no cells were attached to the node. And first we see a huge trunk with 59 copy number events that were collapsed there. And we have printed the breast cancer genes for each node if they overlap with copy number events that were inferred. And the underlined genes are the genes that are unique, so they only appear once throughout the whole tree. And the colors mean the number of cells that were attached to each node. And basically in the trunk, you can see many copy number events that change the known breast cancer genes, including TP53 and RB1, which are deleted according to our final copy number calling procedure. And what we see later is branching of the tree that divides almost all the cells into two distinct subclones. One, the smaller one on the left, which is characterized by the anti-RK3 unique event, and the bigger one on the right is also characterized by unique event in three breast cancer genes. And here are the results for the same data set of the copy number calling procedure. The upper heat map represents the per bean data, so the corrected counts. In genomic beans that are on the x-axis, and the lower heat map are our inference results. The cells are on the y-axis, and they are in the same order. And white color basically means neutral copy number, red color means amplifications, and blue means deletions. And you can see that CONET denoises the data and infers most of the copy number changes, even the very narrow ones, provided that they are present in an enough number of cells. So in summary, CONET is the first Bayesian probabilistic model that infers jointly evolution of copy number and also calls the copy number in single cells. It is robust to errors in this type of data, thanks to joint modeling of the per bean and per breakpoint data. For simulated data, we obtain excellent breakpoint detection and also the tree structure reconstruction. And we infer plausible events for a true breast cancer dataset. CONET is right now available as a preprint at bioRxiv, but it's almost a year old, and we have done many things. So if you are interested, check out our publication. Should be out soon. And also we have implementation. Just check GitHub CONET and it pops up. And I would like to acknowledge my co-authors, Tomek Cąkała, Błażej Miasojedow, and my PI, Eva, and also our collaborators from Merck and Memorial Sloan Kettering Cancer Center. We have great lab in Warsaw, and we are looking for postdocs. Thank you.
So thanks first for the nice talk and the nice tool. So I assume the labels of the nodes are not unique, right? The events?
One exact event can only happen once. They can overlap, the start and then end can repeat many times, but one event can only appear once. You are not satisfied with my answer.
It's a little bit like the infinite sign assumption, then, isn't it? That the event can just happen on this exact event can just happen on one part of the tree and not in another.
Yeah, but copy number change can happen many times. In one bean, many times.
So you could have, let's say, the same chromosome, and then you have copy 2 to 3 and then 3 to 4.
Not exactly the same chromosome, but first the whole chromosome and then parts of the chromosome.
But that could happen. You could have, let's say, chromosome in a case. Stuff.
Yes, but then our data would just say that there was amplification between the beginning and end of chromosome. We don't have right now, our model doesn't differentiate between those two situations.
So Magda, the phylogeny you showed us, how many breakpoints was that based on? And how many candidates?
That was a lot. I think it was over 1,000.
There was 1,000 breakpoints.
Potential breakpoints.
Right. And of those, how many did you use?
This I cannot say, but because we had 200 events, so at most 400, probably less because some of them repeated, and we have inferred only 200 events. So most of them were dropped out, and we this was our exemplary project, so we wanted to include more. Maybe the inferencing procedure would take a little longer, but then we are sure that we don't miss any breakpoints. And just those that are not relevant, they just don't appear in the final tree.
Copy number changes can sometimes be broad over a long range and not that many. And sometimes you can get the little peaks, which say HER2 or whatever, where you kind of is there a trade-off in the method for detecting both of those instances? Or does it do equally well with both?
No, obviously the more evidence for a copy number change between adjacent beans, the more certain we are that this breakpoint is going to appear in the final tree, right? So if the copy number change is higher, then probably our model will include it with more certainty, because data will demonstrate it better. If the copy number changes by 1 due to the noise in the data and the low coverage, this may somehow be dropped out, right? And also what we do is we use, in terms of the length of the events, we use priors. So you can somehow tell the model that you expect shorter events more often than the long ones.
So how many bins do you start out with? When you say candidate breakpoints, is that the total number of adjacent bins, or is it a selection from those? So do you go in 2 steps? First you start out with the bins, then you get the candidate breakpoints, and then you select breakpoints among those?
We start, what we do is we start with the whole genome, and this is our, let's say, this corrected counts, the per bin data, and we use this whole matrix for regularization during the sampling procedure. So we check whether in the places where the copy number change event is, our model tells us that's the same copy number change history, whether all the bins inside there have more or less similar corrected count. But for checking the probability of a breakpoint, we first just establish the breakpoint loci using a different method, for example, HMM copy or CBS plus merge levels. And then we only leave the data in this specific potential breakpoints. So over 1,000 breakpoints times 260 cells.
Right, but how many bins do you start?
To the left and to the right, you mean? No.
Where you potentially detect breakpoints?
Oh, when we potentially detect, we go look through the whole genome.
And how many is that?
Well, you need to divide 3 milliards, but that would be...
bin size?
So 150 kilo.
That's about, so 500 kb is 5,000, so then, I don't know, a third of that, or 3 times 15,000.
Yeah. So a lot.
So one question, when you simulate an end, no? A beginning and an end of the event.
Yes.
But you simulate bins, so they can only occur within a bin in your simulation.
They occur, our simulation is saying that they occur in all the bins between the start and an end.
Okay, so it can overlap multiple bins.
Yes. Yes, the events can be as wide as we want, and they can also overlap. So yeah, we can see that. Sorry?
With each other.
Yes.
You said, sorry.
So you compared your method to other methods in terms of precision and recall. For the 2 steps, so the breakpoint detection is one thing, and then constructing the tree is the second step. So what are the cases when your method outperforms the others, and what are the cases when your method also gets suboptimal results?
In terms of breakpoint detection, sometimes, because we had more scenarios, of course, and Sometimes the methods that only perform copy number calling perform similarly to ours. And in terms of the tree reconstruction, it's only like one method, and we are always much better than the other one. But obviously, this inferring the edges, the exact edges, so the start and end of two different events, and they should be together in the tree. So this is much more difficult. And this here we like have 70% precision, but it's maintained across all the scenarios.
What do you mean by much better? Because you have simulated data and you know the ground truth, but how different the trees can be?
Yeah, here you have the results, like comparing two copy number event trees like by one measure is very difficult because you don't have even the set of all the nodes. You can infer smaller or larger trees with completely different sets of labels. So we have done that, and we have also simulated the relationship between children and parents. And here we also perform better, but...
So you don't have a standard
method to compare? No, this is not.
So you only look at specific factors.
Exactly. Yes, because we don't like the trees are very specific, and we don't have a measure to compare them, like one measure. No.
One more question maybe. So you say low noise and high noise.
Mm-hmm.
But so first of all, what's that actually? What does it mean in terms?
Yeah, the noise in terms of those two distributions. So we have either higher or lower variance, and in the high noise setup, we also add like every one of like 10, I think, corrected counts is just random.
But in a way, what you want to look at, though, I mean, I think that's related actually, but what you care about is the coverage that you have in the cell, no? Because so basically the number of reads you get in a given cell, and you have also normalized for diploidy, because that will tell you the difference between the states.
Yeah, but
the mean, and that's ultimately what gives you the quality of your segmentation.
Okay, but we only simulate the corrected counts, already corrected counts, and so this gray data on the left, we simulate that, and then we, with high noise, we just make...
This would be your standard deviation, I guess.
Yes, yes.
So you don't really, so when I ask you now as an experimental person, how many reads do I want for this thing, how would you get that from that data directly? Is it like...
We don't have a method to do that. No. In our model, we assume we start from the corrected counts. And obviously, what's interested to us is probably the higher the coverage, the less variance should be in the data. The data should be cleaner, but we are only interested in this.
Okay, good. Any other more questions? Then thank you again.